something else that influences, that interests undergraduates usually. So this is looking at alcohol behaviours, and we actually did this study where we got twins, and we got 200 pairs of twins in on a Saturday morning, and not all at once I should say, and got them drunk on a large dose of alcohol, about the equivalent of a bottle of wine, which they had to drink in 20 minutes on an empty stomach. And then they underwent this 3 hours of fairly vigorous psychomotor testing, sort of simulating driving skills, coordination and reaction speed and things like that. And at frequent intervals, we measured their blood alcohol levels, which you see down in this bottom right panel. If the thing would come on. Yeah, and so we're able to look at various aspects of their behaviour, but this is just looking at their alcohol consumption, and showing that in fact there's a remarkably large genetic contribution to how to what your blood alcohol content is and how you handle alcohol, both in terms of the peak alcohol content that you reach and the rate at which you get rid of it. And that's not surprising, given that we know that there is variation in the main alcohol metabolizing enzymes. But that also translates into alcoholism itself, and where we find that for both males and females, this is just that about 2/3 of the variation in the risk of becoming an alcoholic is due to genetic factors. Almost nothing due to home environment, shared environment. That was really quite a surprising result, and the first time that it had been found. So another one of my major research interests is actually trying to find those particular genes that influence alcoholism. I'm interested in behaviour, or behaviour originates in the brain, and so we started this study in 1996 to go directly to take this head on, as it were. Sorry for the pun. I've never actually said that one before, and do this very extensive brain scanning on our twins. So we get them into the Wesley Hospital, where there's this very powerful brain imaging tube, 4 Tesla for those, and we do structural, functional MRI, and we also look at connectivity using a method called diffusion tensor imaging. This is a pair of twins who were once rock stars, so called the Veronicas. Does anyone know them? Yeah, so they took part in our study and allowed us to use their picture for a promo. And in fact, yeah, we've actually, this slide's a bit out of date, we're up to 1,200 pairs now that we've scanned, but and we're looking at all aspects of the brain, but this is just one where we're looking at the brain structure, and it's a heat map of the brain, and the lighter the colour, the warmer the colour, the higher the genetic influence. It's quite interesting. You see that on the cortex, that's the surface area of the brain, the genetic influences are actually quite small, but once you get to the subcortical structures down here, underneath the cortex, you find very, like the hippocampus and nucleus accumbens and so on, you actually find very large influences of genes on the structure of the brain. And these are of great interest, these regions, because these are the regions that tend to be most associated with psychiatric disease, like depression and schizophrenia, bipolar, and so on. So we've got a very large research program investigating the relationship between brain structure and psychiatric disease and the genetic influences on both. Now, all the way through, I've emphasised the similarity of identical twins and comparing them with non-identical twins, but there's also great influence, great interest in just looking at identical twins, and particularly those that are discordant. If they are genetically identical, how come they're discordant? Well, it's the environment. Yeah, but what do you mean by the environment? You know, what does that actually boil down to? How can we use that? So there's a large interest in trying to find these pairs who are discordant and try and figure out what it is that could be making them discordant. And so, I mean, these typically split into two kinds of study where you actually find twins who we know have been exposed to different things and see what the effect of that is, or we start off by observing that they are different and trying to find out what could have caused it. So, try and guess which one of those smoked. Who thinks it's the one on the right? Yeah, you got the point, yeah. So that's quite dramatic, isn't it? So this is a bit more subtle. I mean, this enables you to test sort of hypotheses that take off out there in the ether with very little idea. So evidence. So there was this whole scare that anaesthesia might lead to learning disabilities. And my colleagues in Holland did this very neat study where they were actually, from their huge twin register, managed to identify twin pairs where one had had anaesthesia and the other hadn't, and they then looked at their educational attainment and at their cognitive problems. Is there any cause for concern? None whatever, absolutely none whatever. And in fact, if anything, the ones who were non-exposed to anaesthesia did slightly worse in terms of cognitive problems than the others. But in terms of educational attainment, there is actually no difference whatever between the one who had an anaesthetic and the one who didn't. There's also a debate raging about whether it's better for girls and boys to have a male teacher or a female teacher, particularly in primary school. And they were actually able to find pairs of identical twins who had been split between classes, where one had a male teacher and the other had a female teacher. And those are the results. You know, basically there's not much in it. You know, I mean, it looks as if both girls and boys did slightly better with male teachers. The boys maybe slightly more, maybe a slight interaction effect there. But if it's there, you have to say it's a very small effect. But it's just an example of how being clever, having a large twin registry, being able to pull out these rare subsets of twins, and being clever in the way you ask the question can enable you to do a really incisive study that just can either prove a hypothesis or knock it on the head, as in the case of these two examples. So to take an example of the other, discordant for phenotype. So this is a study, I almost finished now. This is a study we did about 10 years ago, less than 10 years ago. This is a pair of twins, identical girls, where one was completely normal, but the other was born with this really appalling spinal malformation, a duplicated spinal cord. And this reminded the researchers of a mouse phenotype where you actually see a duplicated tail in the mice, and this has been shown to be due to a mutation in a gene called axon 1. And so the first thing the researchers did was to actually sequence that gene, axon 1, in humans and look for mutations, but they couldn't find any at all. So that then led them to think, well, maybe there's something else going on. Maybe it's epigenetic. And so what they did was, now, who knows about epigenetics? Who knows about methylation? Who doesn't know anything about it? You don't. Do you reckon I should explain it all just for her? It's so boring. Okay, so here's DNA, right? And you've got 4 letters, A, C, T, G. You know that? Adenine, cytosine, guanine, thymine, all right? Now, in fact, to the cytosine, just one of those 4 bases, sometimes you can get a methyl group out of it, CH3 group. And if you get a lot of those methylated cytosines in a gene, particularly in the promoter upstream controlling region of a gene, it closes that gene down. So it doesn't function. And it's thought that this is the main mechanism, one of the main mechanisms by which genes are regulated. So, I mean, so the interesting question when we find this pair of identical twins who have got this vastly different phenotype is, could this be caused by different methylation patterns, and in particular, different methylation patterns in this gene axon 1. So we went in and did methylation typing in the promoter region of this axon 1 gene in these two twins. And every circle here represents a cytosine in the promoter, and if it's dark blue, that means it's methylated, and if it's light blue, it means it's not methylated. And you've really only got to look at that to sort of get a feel for what's going on. I mean, it's very interesting because what you see is that both twins are much more methylated than controls. But the affected twin is more methylated than the unaffected twin. You have to, if you can't see it, you just have to believe me. We've done a significance test. And that's sort of interesting because that's telling you two things. Firstly, that probably there's a genetic predisposition to this being more methylated because they're both more methylated than the controls. But some sort of freak stochastic event, some small accident of development has just pushed this one a little bit more methylated than that one, and that's enough to tip this one over into having this horrible spinal malformation, but leave this one normal. So it really illustrates the point that both genes and environment, in this case probably some stochastic developmental effect, have produced it. Luke?
What are the controls?
Well, these are the controls. These are normal twins. So, I mean, one of the obvious questions is this something freakish about being twins, identical twins? So we looked at more just normal identical twins as controls. Yeah. But now there's only one case of this, so you might say, well, what's the point? It's only one case, who cares in the greater scheme of things? But we think it's a paradigm as to what might be causing discordance for all sorts of other diseases, all sorts of common diseases, like, I mean, so people do twin studies of everything: schizophrenia, bipolar, asthma, Parkinson's, you name it. And typically what you find is there's very much higher concordance of monozygotic twins than dizygotic twins, but you never find more than about 50% concordance. 50% of them are discordant. And we reckon that it's mechanisms like this which are causing the discordance. Now, why does that matter? That matters because, not because we particularly care about identical twins, but identical twins are the flag as to what's going on in the population. So we talk about genes and environment being important, and why does the disease suddenly crop up in a family where no one's seen it before? People think, oh, it's just some freakish environmental effect, but we think it's very likely to be this sort of freak sort of change of methylation status for reasons that we currently have no idea what the cause is, and I suspect we probably will never really find particular environmental triggers for these. I mean, for some we will. I mean, certainly smoking, lung cancer, we know that's a big one, but for most of these, I suspect it's just stochastic, it's just errors of development. Right, I think I've spoken for about long enough. I was just up with my plug. Any of you like this stuff? I love having bright students. It really pains me that nearly all my bright students have to import from Europe or Mexico or somewhere. I don't think I've got a single Australian PhD student at the moment. I don't know why. They're all off earning more money doing law or something. But if this does turn you on, and I would say, I mean, it's great if you've got some genetics, genomics. It's great if you at least don't hate maths. Even better if you like it. Good if you, if you like statistics. You've got to be into computing. And I think most of all, you've got to have an interest in the human condition in its broadest sense. Just be fascinated by why people are so different from each other, and what causes that, and exploring that, and then of course in the long term, actually perhaps being able to find therapies that might actually enable us to prevent disease. Because, as I always say, if your car's bung, a very good first step in fixing it up is to find out what's actually wrong with it. Instead of just throwing some WD-40 into the spark plugs or something, or whatever you do, it's a good idea to actually find out what the trouble is, and then you might be able to do something about it. And just to say that we're very pleased to have students out at QRMR.